Good morning, everyone. We have a thesis defense today by Mr. Yu-Chen Chen. And Yu-Chen, even though still just getting his PhD, already have a very interesting academic educational path. He was originally trained as a medical doctor and practiced medicine in Taipei for 2 years, but so attracted to mathematics and applied mathematics, he decided to switch the field completely and started as a master's student at University of University of Michigan, and went on to spend some time at UCLA and then joined our graduate program. So I have to say, I'm very pleased with Yu-Chen's transition. In fact, Yu-Chen was the first of my students to really write a thesis containing some mathematical theorem and got published in a respected mathematical journal, mathematical physics journal. And he has more interesting things he wants to do, but I guess that's going to come later, applying these mathematical results to real biology. So the talk today is going to be the asymptotic behavior and the perturbation analysis of stochastic dynamics and application to complex systems. So Yu-Chen, go ahead.
Chen, thank you for your introduction. And I want to thank you everyone to join my presentation today. So this is a topic Hong has introduced to you. It's about the asymptotic behaviors and perturbation analysis of stochastic dynamics and application to complex systems. So first of all, I would like to introduce what is complex system. So this is about the hierarchical structure. And this slide is from my GSR Professor Healy. So hierarchical structure, this concept prevails among scientific point of view on complex system. And you can see these level structures. And those levels are defined by different scales. So for one level, it has very large scale compared to its lower level. So it is a complex system in relative to its lower level. So let me use this organ as an example. So for this organ, at this level, if we have one organ, it seems a very simple system, just one. However, if we examine this closer, then we might see a lot of cells. One organ might consist of a trillion of cells. So obviously, it's a very complicated system. And you might heard about many-body problem from mathematics. So from this analogy, even 3 cells, this system is already a difficult problem. So how is it possible? We can find some simple law to describe the physiological function for the organ for this very, very complex system. And the answer is actually mathematician has already had an answer for a long time. This is called the law of large number. When you have a huge amount of individuals and the collective behavior of those individuals eventually will have some emergent law. And that emergent law in the limit, when the number of cells goes to almost infinity, you will be able to observe some universal law for the organ, for the higher level. Okay, so this emergent law, this concept was originally from P.W. Anderson. He is an American theoretical physicist and Nobel laureate. And my research was inspired a lot by his paper, More is Different from Science in 1972. And I particularly like this sentence from his paper. At each level of complexity, entirely new properties appear, and the understanding of the new behaviors requires research, which he thinks is as fundamental in its nature as any other. Okay, so today, I would like to introduce you 2 examples. Actually, we can use some very abstract ideas, very abstract concepts we learn from mathematics about the limit law, about the sequence, about the convergence, and we can eventually apply those abstract concepts to justify or to explain some universal law in natural science. And the first example is about asymptotic behaviors of conditional probability. And later, I will use this asymptotic analysis for the conditional probability to explain and justify the canonical ensemble theory. And this example is based on those 3 papers. Okay, so first of all, I want to introduce the canonical ensemble theory. This is from the statistical mechanics. What is canonical ensemble theory? This theory is one to justify the distribution of a small open system, which is contained in a large isolated system. It sounds a little bit abstract. So let me give you the simple example to illustrate this theory. Imagine you have an RNA, RNA is a macromolecule, but compared to a tank of solution, it is much smaller. And you put this RNA into this solution, and then you assume the total system means this big box is isolated, which means it cannot exchange the energy with outside. This is not allowed. So under this assumption, we know that the total system has a conserved energy. However, for the RNA, this small system, since the boundary is free between the RNA and the solution, so we allow the energy can flow in between those 2 systems by this free boundary. So what do we expect? First of all, we will expect to see the energy fluctuations because this is an open system for the RNA itself. Furthermore, we are also interested in the microstate of RNA. And later, I will give you more formal definition about microstate from the statistical mechanics. But here, you can simply imagine this macrostate is the shape of RNA. Each RNA is a macromolecule, so it has very high degree of freedom. So when you put this system under the microscope, you take a picture of the RNA, you might observe different shapes of RNA, like the picture I drew here. You might observe the coil shape or the circle or this very weird shape. So the other question is, what's the distribution law for these shapes? Means the microstate. So now the canonical ensemble theory is trying to answer 2 distributions. One is the energy distribution for the RNA. The other one is the microstate distribution for the RNA. Surprisingly, physicists, they observe some very simple universal law is the distribution for the microstate, this first one, just followed by the very simple distribution, that is exponential distribution. Okay, and in what sense? It's in terms of the energy. Okay, so which means if I'm interested in what's the probability to find this state of RNA, I have the microstate, right? This microstate of RNA. Then I put this microstate into the function called the energy function U, this U function. Then I know the energy of last state of RNA. And this energy will tell us how likely I will be able to find this microstate. If energy is higher, is high energy, then I'm less likely to find this state of RNA. And this less likely is decay in the exponential way. So this is the decay exponential function, and it's controlled by this key parameter beta. This beta controls the decaying rate. And the other interesting result from statistical mechanics is this beta, this parameter, is uniquely determined by the solution. And so I will call this solution heat bath or temperature bath, which means now you can put RNA or any other kind of molecule like protein or DNA. If they are small enough, then you are always able to observe this universal law, this exponential distribution. And this exponential distribution is uniquely determined by the parameter is uniquely determined by that solution. If we fix one type of solution. So it's amazing. It doesn't matter about what the molecules you put in, you always have this universal law, and it's always exponentially distributed. And it's called the Boltzmann law. Furthermore, what's the distribution for the energy? And this is very straightforward, because for one energy, okay, take this example, the red energy. This red energy might have different microstates. And in this case, those 5 microstates, they all have the same energy. And this is called the degeneracy. So which means if we know the distribution for the microstate, then we can further weight by that degeneracy 5, for example, this red energy. Then we know the distribution for the red energy. That will be this exponential times 5, the degeneracy. Okay, so this is the conclusion. Canonical ensemble theory actually tells about two universal laws. One is the microstate follow very simple exponential distribution. And the energy distribution law is an exponential distribution weighted by the degeneracy. And the other important information is that parameter is uniquely determined by the solution. Okay, and what's the goal today? The goal, we want to prove those two distribution law from very mathematical point of view. So this is the goal. First, we want to justify the distribution of energy. And we also want to justify this distribution law for the Boltzmann distribution for the microstate. But here I want to emphasize, we want to justify those two laws for heterogeneous system. What does this mean? Actually, this system is exactly like this example, the RNA system. The RNA has different nature compared with its solution. So this system is defined by heterogeneous system. So on the other hand, there's another system called a homogeneous system, which means the small system, this system of interest is exactly the same one compared to its solution. And the simplest example is ideal gas. You have a box of gas and you are interested in what's the distribution of the small box, the subsystem inside this ideal gas system, and that is homogeneous system. And the Boltzmann law has been justified through the other way for the homogeneous system. So here I want to focus on the heterogeneous system. And the truth is the Boltzmann law and the energy distribution law for a heterogeneous system has been widely checked and observed in biophysics and biochemistry. But the mathematical proof, the justifications are still missing. And the last question is, we want to further answer whether or not we can find the Boltzmann law for the strong couple system. And later I will explain with more details what does the weak interaction and the strong interaction mean in this setup. Means the RNA has very strong interaction with some kind of solution. And the other situation is you put RNA into the other kind of solution and the interaction is very strong. And the Boltzmann law in that scenario with the strong interaction is still the open question. So this is the last question I will answer. Okay, so inspired by mathematical theorems, the law of large number, central limit theorem, we know if we take a sequence of RID, the sum of sequence of RID, and if we scale properly, eventually we will observe some simple law. And law of large number tells you in a limit, you will observe some deterministic number, and that is mean. And the central limit theorem, if you scale properly and shift to the mean and divide by square root of n, you will see a bell shape, and that is normal distribution. So in the large deviation principle, I will talk a little bit more about this one in my second part of the talk. So inspired by those limit theorem, we might have an idea. That exponential distribution in the Boltzmann law and the energy distribution, this might be a consequence of certain limit. And if we can translate that physics setup properly into the mathematical limit theorem, then we might be able to find a new limit theorem beyond those three. And that can help us to justify the Boltzmann law. Also, this is the idea. Let us start with the energy distribution, and later I will show you the microstate distribution. So let us consider this random variable x. X represents the energy of the RNA, and y represents the energy for the solution. And based on that setup, we assume x is much smaller than y because RNA, this molecule, is relatively small to the solution. And the fourth assumption is x plus y is a constant. And this is corresponding to the big isolated system, means the total box is isolated. Okay, and the original problem is asking, under this setup, what's the distribution of the energy for the RNA? And translate this problem into the language in probability theory. It becomes the probability, it becomes the problem of what's the conditional probability of x. Conditional x plus y equals to some constant. So this is the idea. And to translate this idea rigorous to mathematics, we have defined a sequence because we want to show some limit law. So the sequence now are defined by two sequences of random variable. One is xn, the other one is yn. Xn represent the energy for the RNA molecule, and it goes to 0 in the limit. And Yn represent the energy for the solution, and it goes to some finite random variable in the limit. But I want to emphasize this is a relative concept. So which means Xn, the ratio of Xn to the Yn eventually goes to 0. And this represents the first setup. The energy of RNA is relatively small to the solution. And the second assumption is we don't assume Yn is the RID copy of Xn, and this is corresponding to the heterogeneous system. And the third assumption is eventually I want to answer the third question, whether or not the Boltzmann law is still true for the strong interaction system. So we want our setup being more general. So we don't assume X and Y are independent. We allow more general case, they might be dependent. So this is corresponding to a strong coupled system. And the fourth condition is Xn plus Yn is conditioned on some interval. And this is corresponding to the total system is conserved. But we use the interval instead of a number because we want to well define the conditional probability. It has to condition on some non-zero major set. But from the perspective of physics, actually this interval is important because even though we assume the total system is conserved, but due to some uncertain principle from quantum mechanics or measurement, actually this conserve is in the sense of conserved in the energy shield, not the energy surface. Okay, so but for simplicity, you can imagine the total system is conserved in some quantity, but actually we allow a very, very tiny fluctuation for the total energy. Okay, so now the problem is, what's the conditional probability of Xn conditional Xn plus Yn in this small interval when the n goes to infinity? Can we, whether or not we can find a nice asymptotic formula for this conditional probability? And this is the key lemma in our paper. In the limit, we show this conditional probability when n is large has a very good approximation. It can be written as this form. And this Zn is the normalization factor. That is in order to well define, the right-hand side is the density. So the integral is 1. But the most important part is the second two terms. The first one is the unconditional probability, Fxn, and the other one is the exponential weight. Okay, so this result is quite interesting. The conditional probability in the limit can be decoupled to the two functions. One is its unconditional probability. The other one is an exponential weight. And that exponential weight is a consequence of the condition. And this is only true when Xn is much smaller than Yn. So in that limit, we can decouple this conditional probability. And furthermore, we also have the formula for this key parameter for the exponential weight. And it can be written as this function. And later, I will give you more interpretation about this one. Eventually, I will use this one to explain the temperature for the distribution for the Boltzmann distribution law. But here, I can give you some insight. We have these two terms. The first term is solely determined by y, and y is the solution, which means it is the heat bath, the temperature bath. So the first term is uniquely determined by y. So this is corresponding to the physics observation. The temperature of the small system is uniquely determined by the solution. However, we have this correction term. Why? Because we have a more general setup. We don't assume X and Y are independent. And that assumption was true for the classical statistical mechanics. So if the X and Y are independent, then the temperature is uniquely determined by the first term. However, if the system has some dependency, then we have to correct by this term due to the correlation. And I will apply this one later to the strongly interaction system. Okay, so through this lemma, I will justify the distribution law for energy, and then I will justify the Boltzmann law. So as I promised at the beginning, I want to talk a little more about what's the concept of the microstate. Actually, the microstate is one of the points in the phase space. And what is phase space? Phase space, this very abstract concept is actually from statistical mechanics. This phase space helps us to understand all the information about the system. So let us imagine this total system is very large. So it can break down into, for example, 10 to the 23rd. Okay, it's one mole. So it has 10 to the 23rd molecules. And we assume each molecule is the smallest unit. So now for each molecule, if we want to describe the state of each molecule, we need 6 degrees of freedom. If we consider both position and the momentum, and we are in the 3-dimensional world. So X and Y for position, X and Y for momentum. So each molecule, if we have 6 degrees of freedom, we can properly describe the state of each one. But now you have a 10 to the 23rd molecules. So in total, you need a 6 times 2 to the 23rd degree of freedom to describe the total system. Yeah, it sounds crazy, but it's possible. If you have a 6 times 10 to the 23rd dimensional world, then each point is actually can tell you the current state of the total system. And that is the idea of the phase space. So now this point in this gigantic dimension phase space tells you about the current state of the total system. And what does this shell mean? Shell means the energy. Once you know the microstate, and there is a function called the Hamiltonian function, you can use the Hamiltonian function and put all the information about the position or momentum for each molecule. And the Hamiltonian function will tell you what's the energy for that microstate. And why we have a shell? Because we assume the total system has a conserved energy. So now the only possible microstates you can observe for this total system has to be one of the points inside this energy shell. It cannot move, it cannot cross the boundary because we assume the total energy is conserved. However, if we project down this total gigantic system into 2 subspaces, one is for RNA, the other one is for the solution. And now the space for RNA now is much smaller, but it's still very large. So for example, RNA has maybe 6,000, we can break down the RNA into 6,000 units. Then each unit has 6 degrees of freedom. So this space for the RNA will be 6,000. And each point represents the microstate of the RNA. But this is different from the total system in terms of the energy, because now the RNA, the microstate, can move around to everywhere inside this phase space because the RNA itself is open system. So energy can fluctuate. So it doesn't have to be restricted to any energy shell. So the Boltzmann law is actually want to answer what's the distribution law of the microstate in this whole space. What's the distribution law in this phase space? That is the Boltzmann law. And the energy distribution law is about what's the distribution among each energy shell. That's the distribution law for the energy. Okay, so now we have this concept from the phase space to describe what are the laws we are looking for. So we are ready to apply our key lemma to the theorem 1. This one can justify the energy distribution law. So let us start with this big omega space. This is in very high dimension. And in that example, this omega is in 6 times 10 to the 23rd dimension for the total system. And this one can be projected down into 2 subspaces. And one has this vector W1, the other one has this vector W2. W1 is in 6,000 dimension. That is for the microstate of RNA. And the W2 is the microstate for the solution. And on top of this microstate level, we can introduce the Hamiltonian function. Then now we can map this very high dimension phase space into a R1 space. R1 space, since the energy is a real number in R1. So now the problem becomes, we can simplify the problem from enormously high dimension to 1 dimension. And that is the space in the energy. And now we have 2 big assumptions. One is the total energy is given by U1 plus U2. This might sound a little bit trivial, right? You have energy from RNA, you have energy from a solution, and you are asking what's the total energy. It's supposed to be U1 plus U2. But the truth is this is not generally true, because this is only true when we ignore the interaction energy, which means in some scenario, if you put the RNA into some kind of solution, if they have very strong interaction, you can measure some extra energy due to that interaction. But here we assume that interaction is negligible. So the Hamiltonian function follow this additive property. So U1 plus U2 is your total energy. Okay, and what's this corresponding to? Once we have this additive function, then we guarantee that U1 has to be independent to U2, because U1 only depends on the phase space, the microstate of the RNA, and U2 only depends on the microstate of the solution. So those two random variables are independent. Okay, so this is a big assumption. The other one is we know we want the energy of the RNA is much smaller than the energy of the solution. So under those two assumptions, the conditional probability for the energy of the RNA condition on the total energy, this total energy is given by this summation, is conserved in the energy shell. Then we can show in the limit when U1 is much smaller than U2, this conditional probability can be written as this product, and this is the normalization factor, and it becomes the product of an exponential function times its unconditional distribution. Okay, so we almost justify the distribution law by this limit, but there's no degeneracy. We have this unconditional probability. But the truth is, this unconditional probability is proportional to the degeneracy. Okay, you can think in this easy way. If you have an RNA and you put this RNA in a vacuum, means there's no bias, no condition. And what's the most reasonable prior distribution for this, the energy of this RNA in the vacuum? That should be proportional to the degeneracy. The degeneracy means how many states for this inside the one energy shell. So for that energy shell, if that energy shell has more possible microstates, then we have higher probability to find that energy. So that's why the prior energy, the prior distribution, the unconditional distribution, this one is actually the degeneracy. So here we justify the energy distribution law. Then we even have this expression for the temperature, 1 over temperature is this parameter. And the interesting result is this parameter is uniquely determined by the Y, by the heat bath. So this is consistent with physics observation. For this canonical ensemble system, the small system, the RNA, the parameter for this exponential distribution is uniquely determined by the solution. And the correction term disappear. Why? Because now the X and Y are independent. And that is consequence of this additive function assumption. Okay, and we want to further justify the distribution law for the microstate. Right? That theorem tells you about the distribution law for the energy, but how to justify the distribution law for the microstate? Okay, then here, I would like to review this again about this phase space. This is the phase space for the RNA. So far, what do we have? We know the distribution among these energy shells. That is the distribution law for the energy. But now we want to know the distribution law of all those microstates in the phase space. And if the RNA have different states, they might have different energy, right? They might in a different energy shell. However, this is the degeneracy. Those 5 RNA microstates, they might be in the same energy shell because the degeneracy tells us you might be able to find different microstates inside the same energy shell. And this principle is called the equal probability priori. And this is given by some other mathematical theory called the ergodic theory for the Hamiltonian system. Under that theory, we know the probability to find one of these microstates are equally probable, which means each shell is uniform distributed inside the shell. This is called the equal probability priori. With this equal probability priori, this means we have this is proportional to a uniform distribution for inside each energy shell. With this equal probability priori combined with theorem 1, the distribution law for the energy, eventually we can show the distribution law for the microstate is purely exponential. The degeneracy cancel out somewhere, and that is a consequence of the equal probability priori. Okay, so here, on top of the, we combine our result from the energy distribution with the equal probability priori, then we eventually justify the Boltzmann law. That this conditional probability is purely exponential, and this justify the Boltzmann law. Okay, and the last question is I want to answer: can we find the Boltzmann law when the system has strong interaction? And now we have to use this lemma again, because this lemma we have very general setup that x and y can be dependent. In that case, we have a correction term in the parameter of the exponential. Or how to apply this lemma to the strong interaction system? And what does interaction mean? Interaction means you put RNA into the solution. Now the total energy is no longer U1 plus U2. You have an additional term called the U12. And U12 depends on both the microstate of the RNA and the microstate of the solution. And this is called the interaction energy. And now we have a trouble. We have 3 energy, and we have this mutual energy from both systems. So the problem is how to define the energy for the RNA now, because we have this extra term, how to distribute this one properly into the 2 parts. If we simply group the U12 to the U2 and we define the energy for the heat bath, then we have x and y, right? This new y and the x is defined by the original Hamiltonian of the RNA. But now the x and y are no longer independent because y is also depending on the W1 from this interaction energy. Okay, so by our lemma, actually we still have this distribution for the energy. If we modify that parameter has a correction term, we are still able to find the energy distribution law. However, since x and y are no longer independent, we don't have equal probability priori, so we cannot further justify the Boltzmann law. I give you some illustration why the equal probability priori is no longer valid. When the system has no interaction, then we expect those 5 states, the degeneracy, are equally probable inside the energy shell. However, if you have very strong interaction from the solution, and for example, this interaction is some kind of stranging force. Stranging force can strange the RNA molecules. So in this situation, what do you expect about the probability of those 5 states? You are more likely to find this state compared to this state because of the stranging force. So now, even though those 5 microstates have the same energy, but they are no longer equally probable to be found in the energy shell, in the same energy shell. So the equal probability priori is violated due to this strong interaction. So what's the solution? The solution is if we can construct some new Hamiltonian called the pseudo-Hamiltonian to redefine the geometry, then now we guarantee the microstate inside this energy shell defined by the pseudo-Hamiltonian are equally probable. Then we have like equal probability priori, but this is with respect to the pseudo-Hamiltonian. Once we have this pseudo-Hamiltonian, and this is a big assumption, and the truth is in our paper, under certain conditions, we prove the existence of this pseudo-Hamiltonian and find a way to construct this one. Once we have this pseudo-Hamiltonian, then we have the equal probability priori with respect to this pseudo-Hamiltonian. So we can justify the Boltzmann law, but I will call this is a modified Boltzmann law. It's still exponentially distributed. However, it is through the new Hamiltonian, this pseudo-Hamiltonian function. And the parameter, this key parameter has to be modified as well. From the lemma, we know if the 2 systems are not independent, then we have this correction term. And the key function in this correction term is this C function. This is also called the copula density in statistics. And in information theory, actually, this one is intimately related to the mutual information. So this C function represents the correlation between the RNA and the solution. And this correlation is non-negligible, is a consequence of that interaction energy. So now with this interaction energy, the mutual effect information tells you basically the RNA can crosstalk, can talk to the solution due to that interaction energy, and is reflected by this mutual information. So this is the conclusion for my first example. We are able to justify the energy distribution law and the Boltzmann law for the heterogeneous system. And we can even further to answer the question. The Boltzmann law is still true for the strongly coupled system. However, we have to redefine the Hamiltonian by the pseudo-Hamiltonian and correct the parameter for the Boltzmann law, that beta. We have to correct this one with the correlation. Okay, so that's my first example, is using the limit theorem in conditional probability to justify the canonical ensemble theory. Now I want to introduce you a second example, is to use asymptotic analysis in stochastic differential equations. And eventually I will use this limit theory to find the landscape for a dynamical system. And that dynamical system has a stable limit cycle. And this example is based on those three papers. Okay, so first of all, I want to introduce you what's the landscape theory for a dynamical system. Okay, so let us consider a deterministic dynamical system. It is given by dx dt equals to bx. So this bx is the vector field for the velocity. And this is a very general setup for the dynamical system. Then you might ask, can we find some landscape to describe the behavior, the trajectory of these dynamics? And the answer is yes, it's possible in some cases. If this B function, this vector field, can be written as a gradient flow, it's a gradient of some potential U. Then once the B can be written as this gradient form, then we guarantee no matter which initial condition you start, you always going down, follow this potential function and follow the direction of the gradient all the way to the stable fixed point on the bottom. However, in general, B might not be able to written as this gradient form. So it means the U might not exist. So in that case, we are still possible to find the landscape. That is called the Lyapunov function. And the Lyapunov function is a strictly positive function. And the other important property is the negative of gradient Lyapunov function dot with the vector field B is always strictly positive. With this property, we will guarantee the dynamics is always has a monotonic behavior, is always going down, followed by this Hamiltonian function. So this is very powerful. It can predict the trajectory. It's always going down to the bottom of the whale, and that whale is the Lyapunov function. So the first problem is, I know there's a different ways to find this Lyapunov function, but today I will provide one way through the limit theorem to find this Lyapunov function. And the other one is, this is the case when the system has a one stable fixed point. However, we might have a system have a limit cycle. So in that case, can we find the Lyapunov function? If we can, what does it look like? And that's become an interesting problem. On one, the landscape theory is important because usually from our mathematics department, we have a lot of groups. We use the dynamical system to model biological phenomena. And so once we have landscape, we can describe this biological problem from a higher perspective. And for example, like cell differentiation. Once we can find the landscape for this cell differentiation and model by the dynamical system, then we start from some initial condition, means these cells differentiate start with certain type, certain prototype. And then the landscape can predict the destiny of this cell differentiation if this trajectory eventually converge to certain whale, the bottom of the whale. And that bottom is actually the certain type of cell. So we know in the long run, the cell will differentiate to that type. So this landscape can predict this biological behavior. And the limit cycle, since the oscillatory behaviors are ubiquitous in biology as well. And for example, like this cell cycle. So if a dynamical system has a long time limit as a stable limit cycle, then if we can find a landscape for this limit cycle, that can help us to predict this cell cycle biological phenomenon. Okay, so this is a recipe today. I want to eventually I want to find that landscape for a deterministic dynamics, and we have 3 steps. First one is I want to introduce you what's the large deviation principle and what is the large deviation rate function for IIDs, random variables. Then further, we will extend this large deviation principle to a dynamical system. And what is this dynamical system? This dynamical system is based on this deterministic dynamics. This one is we are interested, but we perturb this one with small noise. And now it becomes a random perturbation of the dynamical system. And it can be represented by this stochastic differential equation with a small noise. And this small noise is parameterized by small epsilon. When epsilon go to 0, noise disappear, and it converges to our original problem, the deterministic dynamics. And the amazing result is through this limit, there's an emergent function called the large deviation rate function for the dynamical system. And I will show you how to use that large deviation rate function to construct the landscape of the original problem, the deterministic dynamics. Okay, so those 3 limit theorems, I think the first one and second one, we are already very familiar. And the third one is the large deviation principle. And actually, this one follows from the law of large number. We know we have a sequence of IID, and we scale properly by n. When n goes to infinity, it converges to only one scalar, that is the mu, the expectation of x. So this is a very deterministic behavior in the limit. However, you might ask, what's the probability for other points except the mu? And we know for sure they have probability 0 in the limit. And the large deviation principle is to answer what's the rate of convergence for that probability to the 0. And the answer is they converge to 0 very fast for those rare events. The speed is in the exponential order, like this one. When n goes to infinity, all those probability goes to 0 in this exponential order. And the speed is characterized by this phi function. This tells about that rare event should have probability goes to 0 in an exponential order, how fast. And this is given by this phi function. So this phi function is also called the large deviation rate function. Okay, so let us compare these 3 limit law. The law of large number tells you about this random variable, Sn divided by n has a distribution converge to delta function in the limit. And this delta function, the point is mu. And if you look closer on the scope around this mean, and this scale is 1 over square root of n, then you will observe this normal distribution in the limit. And this is a central limit theorem. And if we're interested about the rare event, they have probability 0. But if we take log and divide by n, that becomes the large deviation rate function, tells about the rate of those rare event goes to 0. And that rate function is given by this large deviation rate function. And it has some nice property. Let us put those 3 limit theorem together. And we'll try to characterize this large deviation rate function. First of all, this rate function is convex, and it is lower semi-continuous. But for simplicity, in our talk, we just assume this function is continuous and differentiable. And those 3 properties are very important about the large deviation function around the mean. First of all, we know this one has to be 0, because this is the only one point has probability 1 in the limit. Right, so the decaying rate is 0. So that's why the mu phi is 0. And this is the only one point, because all the other points are the rare points, are the rare events. So they have to go to 0 in the probability. So this one is the minimum. So that's why the first derivative has to be 0 for this large deviation rate function on the mean. And the third one is the curvature. The curvature around the mean is characterized by the central limit theorem. We put this central limit theorem, this normal distribution together. then this central limit theorem will tell us the curvature is given by 1 over variance. Okay, so I will use those three characteristics several times later about how to characterize this large deviation principle rate function, because eventually I will show you this rate function can tell us about what's the landscape for the dynamics. Okay, but so far, this is just the limit theorem for the ID random variables. So we have to extend those three limit theorems to the dynamics, to the trajectory. Okay, so this, how to apply those three limit theorem to the trajectory is based on this setup. This is the deterministic dynamics we are interested in. We want to find the landscape for this one. And the recipe is actually, we have to do a little bit thing first. We add small noise on top of this deterministic dynamics by this diffusion term. And this is also called a white noise. And this D is the diffusion matrix, give you the weight of this noise on different directions, X, Y, Z. And epsilon is the key parameter. We can tune this epsilon to represent how noisy about this system. If epsilon is very small, means this system is almost deterministic. Okay, so now first of all, we will observe the law of large number, sort of law of large number in this setup. If you take epsilon go to 0, then these random processes converge to a deterministic process. Okay, so this something has randomness converge to a deterministic result. This follows the law of large number. And the other one is the central limit theorem. Central limit theorem, we borrow the idea from the ID sequence. We can shift this solution, means the trajectory x epsilon, properly to the deterministic path and scale by square root of epsilon. So now this Z, this dynamic Z means the local fluctuation around the deterministic path. And when epsilon go to 0, we can expect this Z is sort of like a Gaussian distribution followed by the central limit theorem. And the truth is actually this week we derived this stochastic differential equation for the Z. And the Z is a time inhomogeneous Gaussian process. Okay, and this time inhomogeneous means this Gaussian random variable Z has mean and the covariance both depends on time. They are both function of time. So it's basically the result from the central limit theorem, but since we extend from the random variable to a random processes, so now everything depends on time now. So now this random variable, Gaussian random variable, has time-dependent mean and time-dependent covariance, and those two time-dependent functions can be described by those two differential equations. The mu dt and the sigma dt has to follow these two differential equations. And this is the central limit theorem for the small noise diffusion process. Okay, and we are going to use that central limit theorem to the large deviation rate function. So what is the large deviation rate function for this small noise diffusion process? We have the law of large number. We know these random processes, if we tune the noise goes to 0, it converges to the deterministic path. So if the epsilon is small, very small, but still finite, we can still expect to see some random trajectory. But they are sort of rare events, rare trajectory. You have very small probability to find those trajectories. Okay, so now you are asking, what's the probability? What's the probability of those rare trajectories? So we have to, the first step, we have to transform this SDE to the probability density, which means we want to change our perspective from the trajectory to the perspective of the probability. So this is, actually, we have used this for a long time. You have a random variable, but you have a distribution to describe the random variable. So same here, we just extend to the random processes. You have this SDE, then you transform this perspective to the transition probability density of the process. And this P, xt, is a solution of this PDE. So now this SDE actually can be transformed into a PDE to describe this transition probability. And this PDE is also called the Fokker-Planck equation or the Kolmogorov forward equation. Okay, and what do we expect about this solution P epsilon? the simplest one is we know when epsilon go to 0, these dynamics converge to a deterministic path. So this p epsilon has to converge to some delta function. But the difference is this delta function is a time-dependent delta function. And this delta function tells you about the deterministic dynamics. Okay, so now follow from the idea, we have the large deviation principle for ID sequence. We know this Sn divided by n converge to mean. This is law of large number. Then we have the probability is this exponential form. And we know this large deviation rate function tells about all those rare events converge to 0 in this speed. So following from this idea, for our small diffusion process, we know when epsilon go to 0, it converges to a deterministic path. So there should be a corresponding, this probability can be written as this large deviation principle way to approximate this delta function. And this is the rate function to characterize the probability of those rare trajectories goes to 0 is in this exponential form. But not everything is time dependent because we extend the case from the variable to a process. Okay, so we want to have a better understanding about these large time-dependent large deviation rate function. So it's the same way we did for the ID sequence, we will compare the large deviation principle and the central limit theorem and the law of large number together. Then that will help us to characterize this time-dependent large deviation principle. And this slide is to remind you, this is the result from the ID random variables. So we have a curvature and we know the means on the bottom. And for the random process, is basically you can imagine this is one of the cross-section of the trajectory. Okay, so we will be able to extend this one to a more general case when the random variable is time dependent. This time is a random process. So that's the result. Very similar, everything basically are same. The phi function, the time-dependent large deviation rate function, if you put the deterministic path into this function, it's always 0 because it has probability 1. And the gradient is also 0 because that's the most probable path. So it's on the minimum. And the second derivative means the curvature, and that is given by that covariance we derive from the central limit theorem, and they have the reciprocal relation. And this curvature along the deterministic path should follow this differential equation we derive for the dynamics of the covariance. Okay, so why do I emphasize a lot on this characteristic of the large deviation function? It sort of gives us a landscape, but I would say this not yet, it's not finished yet, but I give you some illustration. We have this deterministic path. That is when epsilon go to 0, it converges to this red path. And now what do we have? We know this path is on the bottom. This has value 0, and they are the minimum. So this has gradient 0. And we also know the curvature, the dynamics of a curvature along this deterministic path. So from the analogy, you can imagine this deterministic path is like a river on the bottom of a valley. Then now we know the curvature around this river. Okay, that's good. That is from the time-dependent large deviation rate function. However, if you move along the river and some information are missing because this river is a flat line on the bottom. So you don't know the speed, and you even don't know you should move forward or backward because it's a flat line. This has gradient 0. Okay, so but however, if we look closer, we have a magnifier. We look closer, actually we can see small hills. Why? Because this is a more general theory for large deviation principle. We were focusing on the leading order term, the 1 over epsilon. But actually, when the leading order term is 0, means you plug a deterministic path. This term is no longer dominant. We have to focus on the second order term. That is something second order term here. That is epsilon independent. And then this is called the WKB method. If we include the second order term, and the physicists have some reason, they put this second order term in the front. But basically, this term is the second order term. That is order 1. This is also called a prefactor. And along the deterministic dynamics, since this term is 0, now the second order term dominates. So if we want to know more about that river, it's go up and down, then we have to characterize the dynamics on this omega function. Okay, so how to do that? We can plug this one into the Fokker-Planck equation, and that is the PDE. So we will have a corresponding PDE for the omega. But now we only care about the dynamics, the deterministic dynamics on the omega. So we can change this Eulerian description to a Lagrangian description to see what happens if we follow the deterministic dynamics x hat t. Then what's the dynamics on this omega? By this transformation, we have this differential equation to tell us about that dynamics. Okay, so this is one conclusion. So far, we have two dynamics. One is the curvature. The other one is the dynamics on less small hills. Okay, so we have those two, and we almost finished. The last step is we have to take time goes to infinity. Now this is still the time-dependent rate function, and this is still the time-dependent prefactor. If we take t goes to infinity, then we can assume that time-dependent large deviation rate function converge to some this one called the invariant rate function, and the time-dependent prefactor converge to an invariant prefactor, and this is guaranteed by the existence of the invariant probability density, because remember, this time-dependent rate function and time-dependent prefactor is given by the expansion of this transition probability. If we assume this invariant density exists when we take t goes to infinity, and that's a one-to-one correspondency, we have the convergence of those two functions. And the answer is actually this phi function, this invariant rate function is a Lyapunov function for the dynamics. It's from this set of equations. And actually this is some result from the Fender-Winter theory, and we refine it a little bit to get these three equations. And these three equations is very powerful. This guarantee the phi function, that time invariant large deviation rate function, is the Lyapunov function for the dynamics. Why? Because this large deviation rate function, first of all, it tells us how to properly decompose the function, the vector field B. And you might remember that B in general is not a gradient flow. However, if we have this invariant rate function, then we can decompose this B into two terms. One is given by the gradient of phi and weighted by the diffusion matrix. And the other part, I call this gamma. And from the second equation, a more interesting result is the gamma is orthogonal to the gradient flow. So this theorem tells you there's an orthogonal decomposition for the vector field for the B. And by these two equations, we can show the dynamics is always going down, follow the phi. So the phi is the Lyapunov function, but this is in the weak sense. We can prove at the end, this phi function, the dynamics on this phi function is non-positive. Okay, the non-positive is in the weak sense because the Lyapunov function requires the strictly negative here. And that equal to 0 will give us a trouble for the limit cycle. Okay, I give you an illustration about this landscape given by the that time invariant rate function. It looks like this Mexican hat. Okay, so this Mexican hat has this center part, and it has a button, and the button has some curvature. Okay, and this is actually the Lyapunov function for the limit cycle. We know there's an unstable point inside the limit cycle. So if you start with some initial condition inside the circle, so you can expect the trajectory, you will be pushed away from the center, from the unstable point. And you will have some spiral trajectory all the way to the bottom of the ring. That is the limit cycle. So this head shape is actually characterized the trajectory in this landscape perspective. And that decomposition actually tells about the trajectory has to be spiral, because we can decompose into two parts. One is the gamma, is the circular motion part, and this part follow the gradient. So combine these two vectors, we can expect the dynamics follow some spiral form and all the way to the bottom and join the limit cycle. However, if we only have this Lyapunov function, some informations are missing. We don't know about, we want to characterize the curvature. The curvature is important because that tells about the tendency to join the limit cycle. And the other one is, if we are exactly on the limit cycle, you are lost, because that is a flat ring. You don't know you should go forward or backward, and you don't know the speed difference. And the truth is, a limit cycle, different points, they might have different speed. But the landscape, this is too coarse. We are not be able to find those information. So now that's the problem. We want to know about the curvature along the limit cycle, and we want to use this magnifier to see the small hills on the limit cycle. And remember, we have those two results from the curvature and the prefactor from the transient state, and we take t goes to infinity. Eventually, we can show those two dynamics. This one is for the curvature along the limit cycle. This one is the hills on the limit cycle. They're followed by those two dynamics. And I think, yeah, so that's the conclusion, but there's a lot of interesting story actually behind those two dynamics. Tells about why some point on the limit cycle is faster and why some point on the limit cycle is slower, and why we should go to that way instead of the other way. It all can be answered by these two equations. And this is the conclusion. Conclusion is about the recipe. If you have this dynamical system, how to find the diagonal function? First step is to perturb this one by a small noise with this white noise term. And you take epsilon go to 0, then you will be able to find the large deviation rate function, time-dependent one. Then the next step is to take t goes to infinity. Then the diagonal function will show up. But if the system, this one only have one stable fixed point, then that's enough, because you know this diagonal function tells you the dynamics eventually will hit that fixed point. However, if this dynamics has a limit cycle, then this diagonal function is too coarse. It's not enough. So we need a prefactor, and we need to know the curvature along the deterministic path. That will help us to have a better understanding about the behavior of limit cycle around the limit cycle and on the limit cycle. Okay, and thank you all. That's all my presentation, and that's all the references I used today. And I want to thank my committee, Qian and Professor Berzi, Professor Lorig, and Professor Healy, my GSR, and all the collaborators for those papers. And I particularly want to thank Qian's group. They gave me a lot of valuable suggestions in my dissertation. Okay, so now I'm happy to take any question.
Okay. Okay. So now the public portion of questions. Anyone, please? Yeah, you can just turn on your speak and ask. Or you can type in the index if you're shy. Nobody ask a question? I guess you can explain.
Oh, sorry, I didn't. It is a bad connection.
Oh, I said if there's no question, that must be means that you can give a pretty good, explain everything really well.
Oh. Okay, thank you. I'm glad to hear that.
Okay, so now if we don't have any question, then let's close up and committee members stay.
Thank you, everyone. Thank you, everyone.
Thank you for coming.